genetic disease. It is known as trichinosis, and it affects the health of human and a large number of domestic and wild animal. ଆମେ ସେଥିପାଇଁ ୟାକୁ କହୁଛେ ଜୁନେଟିକ ଡିଜିଜ। ଏଇଟା ଆମର ଡିଫରେଣ୍ଟ ଯୋଉ ଆମର ଅଛନ୍ତି ଡୋମେଷ୍ଟିକ ଆଣ୍ଡ ୱାଇଲ୍ଡ ଆନିମଲ ରୁ ଏଇଟା ହ୍ୟୁମାନ କୁ ସ୍ପ୍ରେଡ କରେ। This disease trichinosis was reported from Europe and United States of America towards the end of 19th century. 19th century रे first trichinosis disease detect ହେଇଛି। और the definitive host for trichinella spiralis is a pig. Pig is the definitive host for this species, which serve as the reservoir host for human. Human ହେଲା reservoir host for this trichinella spiralis species. This spiralis requires only one host to complete the life cycle. It only it completes its life cycle in one host, that is peak. Another host is required only for maintenance and permutation. ଏଇଟା ଆମେ ଦେଖିବା କେମିତି ଆମେ ଯେତେବେଳେ ଲାଇଫ ସାଇକେଲ ଦେଖିବା କେମିତି ଟ୍ରାଇକିନାଲ ସ୍ପେସିସ୍ ରିଜର୍ଭର ହୋଷ୍ଟ ଭାବେ ହ୍ୟୁମାନ କୁ ୟୁଜ କରୁଛି and life cycle are କେମିତି pig ରେ ସେ continue କରୁଛି। Pig, rat or man are definitive host। ଏଠି ସେମାନେ definitive host or of trichinella spiralis। No intermediate host is required। ଆ ପାଇଁ no intermediate host ଦରକାର ନାହିଁ। However, the parasite entering human cannot complete the life cycle and needs another animal for continuance। କାରଣ ଯେହେତୁ ଏଇଟା ରିଜର୍ଭର ହୋଷ୍ଟ ହ୍ୟୁମାନ ବିଙ୍ଗ, ଏଠି ତାର ଲାଇଫ ସାଇକେଲ କମ୍ପ୍ଲିଟ ହେଇପାରେନି। Pigs are pig serves as also a reservoir host for man। ତ ଯଦି ଆମେ ଦେଖିବା ଲାଇଫ ସାଇକେଲ କୁ ଟ୍ରାଇକୋନେଲା ସ୍ପିସିସ ର, ତାହେଲେ କେମିତି ନା ଯେତେବେଳେ ଆମେ ଇଞ୍ଜେଷ୍ଟ କରୁଛୁ ଅଣ୍ଡର କୁକ୍ଡ ମିଟ୍ ଅଫ ପିଗ, ସେଥିରେ କୋଉ ସ୍ପିସିସ୍ ଆସୁଛି ନା ଆମର ଏଇ ଟ୍ରାଇକୋନେଲା ସ୍ପିସିସ୍ ଆସୁଛି। ଏଇଟା ଯଦି ଆମର ହ୍ୟୁମାନ ଷ୍ଟୋମାକ ଦେଖିବା, ଦ ଲାର୍ଭା ଆର୍ ଇଞ୍ଜେଷ୍ଟେଡ ଇନ୍ ର ଅର୍ ଅଣ୍ଡର କୁକ୍ଡ ମିଟ୍ସ, ପିଗ ରୁ ଆସିଲା। Then this larva are released from nerve cell in stomach। କାରଣ ଏଇ ଯୋଉ ନର୍ଭ ସେଲ୍ ଉପରେ ଯୋଉ କୋଟିଂଟା ଅଛି ଆମର ଯୋଉ ଷ୍ଟୋମାକ ରେ ଏସିଡ ସିକ୍ରିସନ ହବ, ଏଇ ସିଥ୍ ଟାକୁ କଣ କରିବ ନା ବ୍ରେକ୍ କରିଦବ। ବ୍ରେକ୍ କଲେ ଆମର ଏଇ ଯୋଉ ଟ୍ରାଇକୋନେଲା ସ୍ପେସିସ୍ ଅଛି ଇଏ ରିଲିଜ ହେଇଯିବ ଷ୍ଟୋମାକକୁ। ଦେନ୍ ଦ ଲାର୍ଭା ଏଣ୍ଟର ଟୁ ଦ ସ୍ମଲ ଇଣ୍ଟେଷ୍ଟାଇନ। କାରଣ ଯୋଉ ଲାର୍ଭା ଗୁଡାକ ସ୍ମଲ ଇଣ୍ଟେଷ୍ଟାଇନରେ ଯିବେ, କାରଣ ସେଠି ଡାଇରେକ୍ଟ ସେମାନେ ଫୁଡକୁ ଆବଜର୍ଭ କରିବେ। ଦେନ୍ ଦିସ୍ ଲାର୍ଭା ମ୍ୟାଚ୍ୟୁଅର ଇନ୍ ଦ ସ୍ମଲ ଇଣ୍ଟେଷ୍ଟାଇନ, ସେଠି ମ୍ୟାଚୁରେସନ ହବ। ଦେନ୍ the female sheds newborn larva that enter lymph or blood। ଏଇ ଫିମେଲ ଟା କଣ ହବ ନା ତାପରେ ନ୍ୟୁବର୍ନ ଲାର୍ଭା କ୍ରିଏଟ କରେଇବ, ଯୋଉଟା କି ନେକ୍ସଟ ବ୍ଲଡ ଆଉ lymph node କୁ ଏଣ୍ଟ୍ରି କରିବ। ଇଟ୍ କଜେସ୍ ହାର୍ଟ ଫେଲୁଅର ଆଣ୍ଡ ସେରେବ୍ରୋ ସ୍ପାଇନାଲ ଫ୍ଲୁଇଡ କୁ ଯଦି ବ୍ରେନ କୁ ଗଲା, ସିଏନଏସ୍ କୁ ଗଲା, ତାହେଲେ ଇଟ ଡ୍ୟାମେଜେସ୍ ଦ ସିଏନଏସ୍। CNS means central nervous system। Then this new larva, new larva enters into the skeletal muscle। ଯେତେବେଳେ ନ୍ୟୁ ଲାର୍ଭା ଗୁଡାକ ଫିମେଲ ନ୍ୟୁ ବର୍ନ ଲାର୍ଭା କ୍ରିଏଟ କରିବ, ଇଏ ଦେନ୍ ଏଣ୍ଟର କରିବେ କୋଉଠି ନା ହ୍ୟୁମାନ ସ୍କେଲିଟାଲ ମସଲ କୁ। ଦେନ୍ ଲାର୍ଭା ମ୍ୟାଚ୍ୟୁଅର୍ସ ଇନ୍ ମସଲ। ସେ ଲାର୍ଭା ମ୍ୟାଚ୍ୟୁଅର କରିବ କୋଉଠି ନା ମସଲରେ। ଦେନ୍ ନର୍ଭ ସେଲ୍ ଲାର୍ଭା କମ୍ପ୍ଲେକ୍ସ ଫର୍ମ। ସେମାନେ ବି ତାଙ୍କ ବଡି ଚାରିକରେ ଗୋଟେ କଭରିଂ ଅଫ ନର୍ଭ ସେଲ୍ ବନେଇବେ, ଯୋଉଟା ଫିଡିଂ ସେଲ୍ ଫର ଦିସ୍ ଲାର୍ଭା। ଦେନ୍ ଯଦି ଆମେ ଏଇଠି ଦେଖିବା, ଦେନ୍ ଦ ନର୍ମାଲ ରେଡ୍ ଆଣ୍ଡ କ୍ୟାଲସିଫାଇଡ ନର୍ଭ ସେଲ୍ ଲାର୍ଭା କମ୍ପ୍ଲେକ୍ସେସ୍। ଏଇଟା ହେଲା କମ୍ପ୍ଲେକ୍ସେସ୍, ଯୋଉଟା ଉପରେ ଗୋଟା ବାହାର ସାଇଡରେ ନର୍ଭ ସେଲ୍ ବନେଇଛି। ତ ଏଇ ଲାର୍ଭା ଦେନ୍ ଫେସିସ୍ ରେ ରିଲିଜ ହବ। Then this larva, these striated muscles are called nerve cells। then ଯେତେବେଳେ ପିକ୍ ଇଞ୍ଜେଷ୍ଟ କରିବ, ଦେନ୍ ପିକ୍ ରୁ ପୁଣି ଟ୍ରାନ୍ସମିଟ ହବ କୋଉଠିକି ନା ହ୍ୟୁମାନ ବିଙ୍ଗସ୍ କୁ। ଯଦି ଆମେ ଡାଇଗ୍ନୋସିସ୍ ଦେଖିବା, କାହିଁକି ନା ଆମର ଡାଇଗ୍ନୋସିସ୍ ରେ ଆମେ କେମିତି ନା ଯଦି ଷ୍ଟୁଲ ଏକ୍ସାମିନେସନ କରିବା, ଯଦି ଷ୍ଟୁଲ ଏକ୍ସାମିନେସନ ଇନ୍ ୱାନ୍ ଅଫ ଦ ସିମ୍ପଲେଷ୍ଟ ୱେ, ଆମେ ଯୋଉଟା କରିଥିଲେ କି ଷ୍ଟୁଲ ଏକ୍ସାମିନେସନ ଯଦି ମାଇକ୍ରୋସ୍କୋପିକ ଏକ୍ସାମିନେସନ କରିବା, ତାହେଲେ ଆମେ ସ୍ପେସିସ୍ କୁ ସ୍ପାଇରାଲିସ୍ ସ୍ପେସିସ୍ କୁ ଇଜିଲି ଜାଣିପାରିବା, କାହିଁକି ନା ଏଇଟା ଗୋଟେ କଏଲ୍ଡ ଫର୍ମ ରେ ଫର୍ମ ହେଇକି ଥିବ। ଆଉ କଣ ନା ମସଲ ବାୟୋପ୍ସି, କାରଣ ଆଙ୍କର ଏମାନେ ରହିବେ କୋଉଠି ନା ମସଲ ଭିତରେ ଏଣ୍ଟ୍ରି କରୁଛନ୍ତି। ଆମେ ଯଦି ଏଇଠି ଦେଖିବା ଏମାନେ କୋଉଠିକି ଏଣ୍ଟ୍ରି କରିଯିବେ ନା ମସଲ ଭିତରକୁ ଏଣ୍ଟ୍ରି କରି ଯାଉଛନ୍ତି। ଏଇ ଯୋଉ ଲାର୍ଭା ମ୍ୟାଚିଓର୍ସ ଇନ୍ ମସଲ ଭିତରକୁ ଏଣ୍ଟ୍ରି କରି ଯାଉଛନ୍ତି। ତ ସେଥିପାଇଁ ମସଲ ବାୟୋପ୍ସି ନେଲେ ବି ଆମେ ଡିଟେକ୍ଟ କରିପାରିବା।
ସାର୍ 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 ପ୍ରିଭିୟସଲି ଏଥର ଯୋଉ ଅଛି ସାର୍ ସିଲଭାଟିକ ସାଇକେଲ, ସେଇଟା ଟିକେ ସାର୍ ବତେଇବେ ସିଲଭାଟିକ ସାଇକେଲ।
ହଁ, କାହିଁକି ମୁଁ ସେଇଟା ଡେସ୍କ୍ରାଇବ କଲିନି କାରଣ ତମର ଏଇଟା ତମର କୋର୍ସରେ ନାହିଁ। ସେ ଯୋଉ ସିଲଭାଟିକ ସାଇକେଲ ଆଉ ସାଇକେଲ ନାହିଁ, ଖାଲି ଅଛି ଏପିଡେମିଓଲୋଜି, ଇନଫେକ୍ସନ ଆଉ ଡାଇଗ୍ନୋସିସ୍ ଆଉ ଏଇଟା ଅଛି। ଯଦି କହିବ ମୁଁ ଡେସ୍କ୍ରାଇବ କରିଦେବି, କିଛି ଅସୁବିଧା ନାହିଁ।
ନାଇଁ ନାଇଁ ସାର୍ କୋର୍ସରେ ନାହିଁ, ଚଳିବ।
କାରଣ ଅଲଗା ଗୁଡାକ ଆମେ ଯୋଉ ପ୍ରିଭିୟସଲି ଯୋଉ ଗୁଡାକ ପଢୁଥିଲେ ପୁରା ଡିଟେଲ୍ସ ତାର ଥିଲା, ଆମର କେମିତି ଗଟରେ ରହୁଛି କି କୋଉଠି ରହୁଛି, ଲାଇଫ ସାଇକେଲ କଣ୍ଟିନ୍ୟୁଅସ୍ ଥିଲା। ଏଇଟାରେ ଲାଇଫ ସାଇକେଲ ପୁରା ନାହିଁ। ମୁଁ ଖାଲି ଇନଫେକ୍ସନ ଷ୍ଟେଜ ପାଇଁ କି ଏଇଟା ଲାଇଫ ସାଇକେଲ ଟା ଦେଖେଇଛି ତମକୁ, କେମିତି ଇନଫେକ୍ସନ କରୁଛି, କୋଉଟା ଇନଫେକ୍ଟିଭ ଷ୍ଟେଜ ଆଉ କୋଉ ଷ୍ଟେଜରେ ଆମେ ଡିଟେକ୍ସନ କରିପାରିବା, ଡାଇଗ୍ନୋସ୍।
ଆଜ୍ଞା
ସେଥିପାଇଁ ଏଇଟା
ଆଜ୍ଞା।
ଦେଖେଇଛି। ଯଦି ଆମେ ଏଇ ଯୋଉ ଖାଲି ଜଷ୍ଟ କହିଦଉଛି, ଏଇଟା ହେଲା ଡୋମେଷ୍ଟିକ ସାଇକେଲ ଆଉ ସିଲଭାଟିକ ସାଇକେଲ। ମାଉସ ରୁ ଏଇଟା ଡୋମେଷ୍ଟିକ ରେ ଯଦି ପିକ୍ ଯଦି ରହୁଛି ପିକ୍ ମାଉସ ଭିତରେ ହବ, ପିକ୍ ଟୁ ପିକ୍ ନହେଲେ ମାଉସ ଟୁ ମାଉସ। ଯଦି ତମେ ସିଲଭାଟିକ ସାଇକେଲରେ ଦେଖିବା ତାହେଲେ ପିକ୍ ଟୁ ମାଉସ, ମାଉସ ଟୁ ବିୟର, ଏମିତି ଡିଫରେଣ୍ଟ ଅଦର ଅର୍ଗାନିଜମ ଆନିମଲ୍ସ ମାନଙ୍କୁ ବି ଇଏ ଆଫେକ୍ଟ କରିବ, ମାନେ ସ୍ପ୍ରେଡ କରିବ ଏଇ ଯୋଉ ଟ୍ରାଇକନେଲା ସ୍ପେସିସ୍ ଟା। ସେଥିପାଇଁ ୟାକୁ କହୁଛେ ଆମେ ସିଲଭାଟିକ ସାଇକେଲ, ଏଇଟା ହେଲା ଡୋମେଷ୍ଟିକ ସାଇକେଲ। ଡୋମେଷ୍ଟିକ ପିକ୍ ଟୁ ପିକ୍ ନହେଲେ rat ଟୁ rat। ଆଉ କଣ ନେକ୍ସଟ ନା ଆମେ ବ୍ଲଡ ଏକ୍ସାମିନେସନ କରିପାରିବା, କାରଣ ବ୍ଲଡ ଏକ୍ସାମିନେସନରେ ଆମେ ଏଇଠି ଦେଖିଲେ ଫିମେଲ ସେଟ୍ସ ନ୍ୟୁବର୍ନ ଲାର୍ଭା ଦ୍ୟାଟ ଏଣ୍ଟର୍ସ lymph and or blood। ବ୍ଲଡ ଭିତରେ ବି ଆମେ ଦେଖିପାରିବା ଏଇ ଯୋଉ ଇନଫେକ୍ସନ କୁ। ଦେନ୍ ଏକ୍ସରେ, ଏକ୍ସରେ କାହିଁ କରିବା ନା ଏମାନେ ଯଦି ମସଲ ଭିତରେ ରହିଯାଇଥିବେ ତାହେଲେ ଏକ୍ସରେ ଯଦି ଆମେ କରିକି ତାହେଲେ ଇଜିଲି ଦେଖିପାରିବା। ଯଦି ସ୍ପାଇରାଲ ଫର୍ମରେ ଆସିଥିବ ତାହେଲେ ଆମେ ତାକୁ ଇଜିଲି ଡିଟେକ୍ଟ କରିପାରିବା। ଦେନ୍ ନେକ୍ସଟ କଣ ନା ସେରୋଲୋଜିକାଲ ଟେଷ୍ଟ। ସେରୋଲୋଜିକାଲ ଟେଷ୍ଟରେ ଯଦି ଏଗେନଷ୍ଟରେ କିଛି ଆଣ୍ଟିବଡି ପ୍ରଡ୍ୟୁସ ହେଇଥିବ ଆମ ବଡିରେ, ଆଇଜି ଲେବୁଲ ଯଦି ଇନକ୍ରିଜ କରିଥିବ ଆଇଜିଜି ଲେବୁଲ, ତାହେଲେ ଆମେ ସେଇଟାକୁ ଡିଟେକ୍ଟ କରିପାରିବା ସେରୋଲୋଜିକାଲ ଟେଷ୍ଟରେ। ତାହେଲେ କିଛି ଆମ ବଡିରେ ଇନଫେକ୍ସନ ହେଇଛି। ବଟ୍ କନଫର୍ମେଟୋରୀ ଟେଷ୍ଟ ପାଇଁ ଆମକୁ ଷ୍ଟୁଲ ଏକ୍ସାମିନେସନ, ମସଲ ବାୟୋପ୍ସି, ନହେଲେ ଏକ୍ସରେ, ନହେଲେ ବ୍ଲଡ ଏକ୍ସାମିନେସନ କରିବାକୁ ପଡିବ। ଦେନ୍ ସ୍କିନ୍ ଟେଷ୍ଟ, କାରଣ ଏମାନେ ଯେହେତୁ ମସଲ ଭିତରକୁ ଆସି ସ୍କିନ୍ ତଳେ ରହିଯାଆନ୍ତି, ସେଥିପାଇଁ ଇଣ୍ଟ୍ରାଡର୍ମାଲ ଇଞ୍ଜେକ୍ସନ ଯଦି ଆମେ ଦବା 0.1 ml ଅଫ 1/1000 ଡାଇଲ୍ୟୁସନ, ମାନେ ଯଦି 1 ml in 10,000 ml ରେ ଦବା, ତାହେଲେ ଏଇ ଯୋଉ ଇମିଡିଏଟ ଏରିଥ୍ରୋମାଇଟ୍ରିୟସ୍ ପ୍ୟାଚ୍, ମାନେ ଇମିଡିଏଟ ହାଇପର ସେନ୍ସିଟିଭିଟି ରିଆକ୍ସନ ହବ, ଗୋଟେ ସେଠି ପ୍ୟାଚ୍ କ୍ରିଏଟ ହେଇଯିବ। ତ ସେଥିରୁ ବି ଆମେ ଜାଣିପାରିବା ଯେ ଏଇ ଡିଜିଜ ଟା ଟ୍ରାଇକୋନେଲା ସ୍ପାଇରାଲିସିସ୍ ହେଇଛି କି ନାହିଁ। ଯଦି ନେକ୍ସଟ ଦେଖିବା ଇମ୍ୟୁନ ଆଣ୍ଡ ଇମ୍ୟୁନ ଇଭାସନ ମେକାନିଜିମ, ଏଇ ଯୋଉ ଆମର ଅଛି ଟ୍ରାଇକୋନେଲା ସ୍ପେସିସ୍, ଏମାନେ ଯେତେବେଳେ ଫାଷ୍ଟ ଏଣ୍ଟର କରିବେ ବ୍ଲଡ କୁ, ଆମର ଫାଷ୍ଟ ଲାଇନ ଅଫ ଡିଫେନ୍ସ ରେ ମ୍ୟାକ୍ରୋଫାଜେସ୍ ମାନେ ଆକ୍ଟିଭେଟେଡ ହେବେ। କ୍ଲାସିକାଲ ମ୍ୟାକ୍ରୋଫାଜେସ୍ ମାନେ ଏମାନେ ଡିଫରେଣ୍ଟ କାଇଣ୍ଡସ ଅଫ ଇଣ୍ଟରଲ୍ୟୁକିନ୍ସ, ମ୍ୟାକ୍ରୋଫାଜେସ୍ as well as eosinophils ସିକ୍ରେଟ ହେବେ। ଏମାନେ ହେଲେ ଫାଷ୍ଟ ଲାଇନ ଅଫ
Sir,
ଡିଫେନ୍ସ
good
ରେ।
evening, sir. This is Dr. Paningre.
ୟେସ୍ ସାର୍।
Our students are not understood English or what?
ସାର୍ ପିଲା କହିଲେ ଟିକେ ଓଡିଆରେ ସାର୍ ଭଲସେ ବୁଝେଇ ଦିଅନ୍ତୁ, ତ ସେଥିପାଇଁ କି ମୁଁ ଓଡିଆରେ ଟିକେ କହୁଛି।
Students are demanding in Odia.
ୟେସ୍ ସାର୍।
But because, I mean, our language is English, that's why I asking. Okay, fine, go ahead.
ଓକେ ସାର୍।
But preferably you speak in English, okay? Because, I mean, because they will not write in Odia. They have to write in English only.
ସାର୍ ସେମାନେ କହିଲେ ଯେ ଯଦି ଓଡିଆରେ ଟିକେ ଆମକୁ ସାର୍ ବୁଝେଇ ଦେବେ ତାହେଲେ ଆମେ ପଢିକି ଲେଖିପାରିବୁ। ସେଥିପାଇଁ କି ମୁଁ କହିଲି, ନହେଲେ
Then why
ଫାଷ୍ଟ
they are doing, why they are doing MSc Zoology? They should do in Odia. They're supposed to not do in MSc Zoology. They tell them they should do in Odia MA. They should not join in MSc Zoology. Because in our system, we are not teaching in Oriya. So when the
ଓକେ ସାର୍।
students are demanding teaching Oriya, so tell them don't join in IGNO. They should go and join in MA Oriya classes. So that is better. Okay, continue, sir.
ओके when the trichinella species enters into the bloodstream of humans, in the first line of defense, the macrophages and the eosinophils are activated. Then it secretes different interleukins, and these interleukins are elevating the alternative activated macrophages and recruit more number of macrophages as well as eosinophils. Then these eosinophils and macrophages clears the trichinella species. So this is the immunity cycles of human beings. Then if we go to the treatment of trichinella spiralis, human trichon human trichinosis is treated with anti-helminthic drugs such as mebendazole. Previously, as well as we discussed about this anti helminthic drug, that is mebendazole, or these drugs kill the adult worms but have little or no effect on the encapsulated larva in the muscles. These drugs are considered relatively safe. However, because their uh, teratogenic effect, teratogens means it causes mutation in the DNA, then these should not be given to the pregnant woman which is suffer from this trichinella spiralis species. And some corticosteroids like prednisone may be given in case of severe infection to reduce inflammation. If inflammation increases, then pain will happen in human beings. So corticosteroids reduces the pain in human beings. Then if we go for the immune therapy, then studies have been conducted in pigs infected with this trichinella spiralis species, and this species total soluble antigen, the trichinella species ର TS soluble antigen ଅଛି. This treatment show primary immune response and the parasite burden declined in the pig intestine. But this study is in pig but not in human being, but we are particularly targeting the human beings. The fecundity of the adult female worms was also affected. This immunotherapy treatment also had a direct impact on parasite burden in muscle tissue. The damage to the CNS, central nervous system, in the muscular phase of the infection was found to be less than the control animal, that is infected animals, without any immunotherapy treatments. Then similar results were found in murine experimental species, but immunization of rats with different nutritional conditions, that is so decrease parasite burden compared to the controls without any treatment. However, more research within this antigen required for using it a viable vaccine in the future, because this study is only conducted on the pig and murine species but not in human beings. There is no vaccine for the human beings. If we go for the different vaccine strategy, then vaccine provide a long term protection. Effective vaccine also help to reduce eliminate the problem of drug resistance. ୱିପି କାହିଁକି ନା ଆମେ ଏଠି କଣ ଦଉଥିଲେ ନା ଡିଫରେଣ୍ଟ ଆଣ୍ଟି ହେଲମିନ୍ଥିକ ଡ୍ରଗ୍ସ, ଯୋଉଟା ମେବେଣ୍ଡାଜଲ ହେଲା କି ଆଲବେଣ୍ଡାଜଲ ହେଲା, ତ ଇଫ୍ ୟୁ ରିଡ୍ୟୁସ୍ ଦିସ୍ ରେଜିଷ୍ଟାନ୍ସ we should vaccinate the human being. For this vaccination, there is no vaccine. We have to choose different vaccinated strategy to develop the vaccines against this trichinella spiralis species. The effective vaccine have yet to be developed as a preventive measure against the infection of Trichinella spiralis species. This vaccine, first one is live attenuated vaccine. The live attenuated vaccine means what? ना जदि हमें Trichinella spiralis species को जदि heat kill करिबा कि radiation ने ताको मारिकी ताको जदि direct हमें से जो species को हमें human being को infect करेबा ताले ଆମେ ଗୋଟେ ଭ୍ୟାକ୍ସିନ କ୍ୟାଣ୍ଡିଡେଟ୍ ବି ଡେଭଲପ କରିବା, ଯୋଉଟା କି ରିସର୍ଚ୍ଚ ଇଜ ଗୋଇଂ ଅନ୍। ଦେନ୍ ନ୍ୟାଚୁରାଲ ଆଣ୍ଟିଜେନ୍ ଭ୍ୟାକ୍ସିନ, ଯଦି କିଛି ଟ୍ରାଇକୋନେଲା ସ୍ପେସିସ୍ ଏନି ପ୍ରଡ୍ୟୁସେସ୍ ଏନି ଆଣ୍ଟିଜେନ୍ ଏଗେନଷ୍ଟ ହ୍ୟୁମାନ ବିଙ୍ଗସ୍, ତାହେଲେ ଆମେ ତା ଏଗେନଷ୍ଟରେ କିଛି ଆଣ୍ଟିବଡି ବି ତିଆରି କରିପାରିବା। ଆଉ କଣ ନା ରିକମିନାଣ୍ଟ ପ୍ରୋଟିନ୍ ଭ୍ୟାକ୍ସିନ୍ସ। ରିକମିନାଣ୍ଟ ପ୍ରୋଟିନ୍ ଭ୍ୟାକ୍ସିନ ମିନ୍ସ ଆମେ ଯଦି କିଛି ପ୍ଲାଜମିଡ୍ ଡିଏନଏ ୟୁଜ୍ କରିକି ଆମେ ଏଇ ଯୋଉ ଆଣ୍ଟିଜେନ୍ against जो antibody भी तैयारी करी परिबा, that is recombinant protein vaccine. Then DNA vaccines, DNA vaccines हला जेमति हमें कहले recombinant protein vaccine, सेमति DNA କୋଉ ଡିଏନଏ ରେ ଆର ଯୋଉ ଜିନ୍ ଅଛି, ଡିଏନଏ ରେ ଯୋଉ ଜିନ୍ ଅଛି, ସେଇ ଜିନ୍ ଯୋଉ ଆଣ୍ଟିଜେନ୍ ବନଉଛି ତା ଏଗେନଷ୍ଟ ରେ ବି ଆମେ ଚାହିଁଲେ ବ୍ଲକ୍ କରିକି ଆମେ ଡିଏନଏ ଭ୍ୟାକ୍ସିନ ବନେଇ ପାରିବା। ଆଉ କଣ ନା ସିନ୍ଥେଟିକ୍ ପେପ୍ଟାଇଡ ଭ୍ୟାକ୍ସିନ। ସିନ୍ଥେଟିକ୍ ପେପ୍ଟାଇଡ ଭ୍ୟାକ୍ସିନ ମିନ୍ସ କଣ ନା ଆମେ ଏଇ ଯୋଉ ଆଣ୍ଟିଜେନ୍ ତିଆରି କରୁଛୁ ତାକୁ ସିନ୍ଥେସିସ୍ ବି କରିପାରିବା। ଯଦି ତମେ ପଢିଥିବ ଆମର ପେପ୍ଟାଇଡ ସିନ୍ଥେସିସ୍, ଯେମିତି ଆମେ ନର୍ମାଲ ସଲିଡ ଫେଜ୍ ରେ ବି ପେପ୍ଟାଇଡ ସିନ୍ଥେସିସ୍ କରିକି ଡିଫରେଣ୍ଟ ଟାଇପ୍ସ ଅଫ ଭ୍ୟାକ୍ସିନ ଷ୍ଟ୍ରାଟେଜିରେ ଆମେ କାହାକୁ ନା ଟ୍ରାଇକୋନାଲା ସ୍ପାଇରାଲିସିସ୍ କୁ ବ୍ଲକ୍ କରିପାରିବା। ଦିଜ୍ ଆର୍ ଦ ଡିଫରେଣ୍ଟ ଭ୍ୟାକ୍ସିନ ଷ୍ଟ୍ରାଟେଜି। Then next parasite is entamoeba histolytica. যদি আমি entamoeba histolytica କୁ ଦେଖିବା, entamoeba histolytica is a parasite that cause amoebosis. Amoebosis is a chronic amoebic dysentery, acute amoebic it causes chronic amoebic dysentery, acute amoebic dysentery, and also colon ulcer, liver abscesses, splenic abscesses, pulmonary abscesses, and cerebellar amoebasis. Entamoeba histolytica is a monogenetic parasite. Monogenetic parasites means kono na these have only one host, that is human beings. If monogenetic, if human being is a host or apes or monkey, these are called monogenetic parasites host. The parasite lives in the epithelial cell of the mucosa and submucosa of cecum, colon, and sigmoid rectal region of human. It present at mucosa as well as submucosa and cecum and colon region of human beings. And this parasite exists in three distinct forms. One is trophozoite. This is the transparent color. This transparent color is called a trophozoite state, and this is pre-cystic state, and another one is cystic state. This is the quadrinucleated cyst which is infective to the human beings. Then this trophozoite of Entamoeba histolytica is the pre-moving stage that feed on bacteria and other organic contents of the host. These are pathogenic in nature and cause different kinds of amoebasis. If this quadrinucleated cyst is infected to different organs like cecum, colon, submucosal layer, and it causes different amoebasis diseases. ଯଦି ଆମେ ଲାଇଫ ସାଇକେଲ ଅଫ ଏଣ୍ଟାମୋeba ହିଷ୍ଟୋଲିଟିକା କୁ ଯିବା, ତାହେଲେ ଏଇଟା କୋଉଥିରେ ହଉଛି ନା, ଯଦି ଆମେ ଦେଖିବା ଏଣ୍ଟାମୋeba ହିଷ୍ଟୋଲିଟିକା, ଏଇଟା ହବ through drinking of water. Next, it goes to the intestine, and this quadrinucleated cyst moves through the esophagus, stomach, and reaches small intestine, where exocystosis occur. Exocystosis means the cystic layer is removed by the stomach acids. Then and this quadrinucleated state, then it goes to the different organ, like previously we tell that cecum, colon, and sigmoid rectal region of the human beings, and it causes different types of amoebasis. If we go to the pathogenesis, what it causes, then the trophozoite from Entamoeba histolytica is the pathogenic form. It can cause various forms of amoebasis. The trophozoite can be categorized into two types based on the pathogenicity and invasion capability. This trophozoite is found in two states. One is non-invasive trophozoites. The non-invasive trophozoites of Entamoeba histolytica colonize in the epithelial cell of mucosa of the colon and sigmoidal rectal area, causing amoebasis. The non-invasive amoebasis is generally asymptomatic with passage of cyst in the stool. It is usually seen in people with normal and strong immune system. If the human being have strong immune system, this non-invasive trophozoite is asymptomatic in nature. If we go to the invasive trophozoite, the non-invasive trophozoite may sometimes become invasive. These trophozoite penetrate the intestinal mucosa and invade extra-intestinal organ like liver. This cause hepatic amoebasis, then spleen, then lungs, then brain, and cause symptomatic amoebasis in intestine and extra-intestinal organ. These invasive trophozoites then go to penetrate to the intestinal mucosa, and it causes different types of symptomatic amoebasis. Then if we go to the diagnosis of entamoeba histolytica, then first is we go for the fresh stool examination. By microscopic examination, we observe this entamoeba histolytica, um, uh, entamoeba histolytica quadrinuclear C stage uh, under the microscope, and the periodic acid skip stain shows under the microscope of entamoeba histolytica species. And if we go for the sigmoidoscopy or colonoscopy, because it enters into the colon and causes the um, mm, amoebasis, and we go for the biopsy of the ulcer, and can be examined by histopathological study to diagnose these parasites. And another one is serological test, as we previously discuss about. If we uh, uh, found that the immunoglobulin level is high in the blood, then uh, we say that the species is affected to the some parasites, and we can test the then we can confirm by uh, fresh tool examination. Then DNA probes and PCR polymerase chain reaction against this species DNA, and paper test. In paper, stool is exposed to a strip of paper coated with the antibody, and this immunoglobulins, and the parasite will stick to the antibody on the paper. If we uh, put a drop of antibody on one paper, then the stool, diluted stool, is exposed to that paper, then it will bind to that paper, and we confirm that antigen antibody reaction occurs, and we confirm this is the entamoeba histolytica reactions, antigen reactions. If we go to the immunity part, uh, if we if we go to the treatment of entamoeba histolytica, then symptomatic patients with intestinal and extra-intestinal amoebasis and can be treated with different these antihelminthic drugs like metronidazole, tranidazole, ornidazole. These can be treated to this worm, and patients with complicated intestinal amoebasis may be treated with antibacterial drugs to prevent secondary infection of intestinal ulceration, because it causes intestinal ulceration. Then we go for the vaccine strategy. Then we target a entamoeba histolytica antigen, namely recombinant 170 kD galactose, that is N-acetyl D-galactosamine galactose, or galactose N-acetyl glucosamine. A 52 kDa recombinant fusion protein and 260 kDa antigen are being tried out in experimental animals, and this research is going on because this antigen recombinant 170 kD galactose, that is N-acetyl galactosamine, this is treated for vaccine against the entamoeba histolytica species. If we go for the immunity and immune evasion mechanism of entamoeba histolytica, then host immune response to intestinal amoebasis can be non-immune defense mechanism, because or specific immunity. Specific immunity means what? Now, specific immunity means innate or acquired immunity. Or non-immune defense mechanism, what? Now, gastric acid in the stomach forms the first line of defense entering the pathogens, because in stomach the pH is 1.8 to 2, and it will dissolve the different cyst of this entamoeba histolytical species. So this is the non-immune defense mechanism. Then the cell mediated immunities, uh, the cell mediated immunities, T cell or B cell mediated immunity. In this cell mediated immunity, different lymphocytes, that is T lymphocytes or B lymphocytes, are involved to kill the Entamoeba histolytica species by production of different reactive oxygen species and reactive nitrogen species. This kill the Entamoeba histolytica pathogens. Then the next parasites is lymphatic parasites, that is Wuchereria branchopti or Brugia. If we go to see the pictures of this Wuchereria branchopti and Brugeria malbi are the prevalent parasites of lymphatic system of human beings. These are endoparasites and found in lymphatic vessels and lymph nodes of human beings, particularly in groin region. This microfilaria Wuchereria branchoptis species found in the human blood, and this is stained with the gymnostain, as previously we discussed about these gymnostains, and both are the digenetic parasites, means it require two hosts because complete the life cycle. Man is the primary host, while large number of species mosquito belong to the genus Culex, Aedes and Anopheles. This is very important. Act as the secondary host for this Wuchereria branchopti species, and secondary host of Brugueria include Mansonia, Aedes and Anopheles. Brugueria ପାଇଁ ଦରକାର ଅଛି କଣ ନା ଏଇ ଦି ତିନିଟା species important species, that is Mansonia, Aedes and Anopheles species. If we go to the, this is a digenetic parasite, means it has two hosts. If you go to the primary or definitive host, humans serve as the only definitive host of the filaria worms, and second one is a large number of mosquito species, that is Culex, Aedes or Anopheles is the secondary host for this Wuchereria branchopti species. If you go to the life cycle of this Wuchereria branchopti species, first infected mosquito introduce the larval three stage into the skin of the human host, from where they penetrate in the body through the bite wound, and then the larva develop into the adult. um, the larva develop into the adult and inhabit the lymphatics, and adults gives birth to the adult give birth to the microfilaria worm, which migrate into the lymph and blood vessel of the human beings, and then the mosquito ingests these seated microfilaria while sucking the blood from the infected individuals, and this microfilaria lose their teeth a few passes through the wall of the proventriculus and midgut of the mosquito, because midgut of the mosquito and the proventriculus contains different enzymes like trypsin and chymotrypsin. It degrades the sheath of the microfilaria worms, and um, they develop into the L1 larval stage and molt into L3 larva, and this mosquito proboscis through hemocells and infect another human during the blood meal. Then L1 is converted mold into L3, and this it goes to the proboscis of the mosquito and it infect the second human beings. If you go to the pathogenicity, then Wuchereria branchopti causative, the disease is usually asymptomatic because it goes through the lymphatic system. So this is asymptomatic and is without any external symptoms or infection. It causes significant damage to the kidney, lymphatic system and immune system of the body. In case of severe infection, various of pathological symptoms, mild to severe, may be observed. This is the elephantiasis, which is caused by this species Wuchereria branchopti, because it resides in the lymph nodes of the human beings and it causes swelling of the lymph nodes, and this is the pathogenic disease called elephantiasis or elephantiasis edema. Edema means swelling. Then if you go to the detection of these filariaceous worms, This phyllosis worm will be detected by microscopic examination because of this size of the this phyllosis species, and this different types of it secretes different types of um, uh, particles outside his body, and it clearly visible through this microscopic examination. And if we go to the antigenic determination, then the detection of micropylaria antigen, that is circulating filarial antigen, that is CFA. And this CFA can be easily detectable by ELISA, in ELISA method. If we go to the clinical diagnosis in the lymphatic regions, this immunoglobulin E, IgE, this IgE is activated in hypersensitivity reaction, and then this can be detected. Then X-ray diagnosis, through X-ray diagnosis we can also detect these elephantiasis worms. And ultrasound examination also shows this elephantiasis worms. And biopsy we also biopsy of lymph nodes, we can easily detect these worms. We can take the biopsy of these lymph nodes, and we can easily detect the worm. Then, if we go to the host immune response, the host immune response to filaria worms in humans is by T helper TH2 type, and it involves in production of different kinds of cytokines. And these different types of cytokines recruit our different types of phagocytic cells like macrophages, natural killer cells, and it clears these worms. And if we go to the next stage, which if we go to the troponin protein, if we when sequenced, has been found to contain multiple antigenic components which can strengthen the human immune system and impact protection against this filariasis worm. This is important that even small fragment of the this troponin protein can produce immune response against the activity of Wuchereria branchiopti, because this troponin protein is required for the muscle contraction. When the elephantiasis edema is occur, this protein is mm, uh, overexpressed in this Wuchereria branchiopti cases. And if you go to the human beings, also exhibit inherited tendency towards this lymphatic obstruction. This worm causes lymphatic obstruction. Due to this lymphatic obstruction, the elephantiasis edema disease was found. Then this supporting the hypothesis that host genes influences by lymphoedema susceptibility. Scientists also have reported that individuals with low levels of mannose binding lectin may possesses higher micropylaria, suggesting a genetic uh, predisposition. Previously, we discussed about this mannose binding protein in our different complement system, lectin pathway. This mannose binding lectin is activated in this complement system pathway. In addition, a tendency to develop a chronic disease that has demonstrated in individual with polymorphism. Polymorphism means sequential changes in the genes of tumor necrosis factor 2 and endothelin. These two factor, tumor necrosis factor and endothelin, it recruits the macrophages of human beings, and these macrophages clears the this filariasis worms. So this is the host immune system of the human beings. If we go to the treatment of this filariasis, then we use diethyl carbamazine. It is piperazine derivative, and it is the and widely used most common and widely used drug. Uh, this DAC targets cyclooxygenase pathway COX-1 and acts rapidly by stimulating host immune system. To enhance the effect of DEC against microfilaria, nitric oxide has been used as synergistic by some researchers. This nitric oxide direct kill this pathogens, filariasis pathogen, by creating pore in the membrane of this filariasis worm. So it then the water will be goes inside the this filariasis worm, and the death of the filariasis worm will occur. Then we go for ivermectin. Everyone knows about the ivermectin during this COVID time. It lowers the inflammation levels, and when the antihelminthic or any viral load occurs in our body, the immune system quickly react against these pathogens and creates a cytokine storm, and it will elevated the human body temperature. So this ivermectin reduces the temperature of the human beings, like reduces by inflammation. Then we go for the suramin and albendazole. This drug causes the it- this anti-helminthic drug also inhibit the pathogenesis of this filariasis worm. Then we go for the last parasitic worms of subcutaneous tissues, that is Dracunculus. Uh, if we go to the structure of the male and female Dracunculus medinensis, the disease is caused by the female one. And this is the male and female one, which is given the term Draconsiasis by Galen, and this is guinea pig worm is the only helminthic parasite that is spread solely by drinking water, and mostly affects rural, poor, and isolated communities like mm, South Sudan, Ethiopia, and Mali, and rarely on open, stagnant surface water sources as ponds for drinking water. This is a digenetic parasite. Parasite means it requires two hosts to complete its life cycle. The definitive host can be humans, dogs, cats, and other wild animals, while mm, the intermediate host is Meso cyclops, a freshwater microcrustacean copepod. This is the copepod cyclops. Inside this cyclops, this species of Dracunculus um, completes its life cycles. Then human acquire infection by drinking unfiltered water containing copepods infected larval stages. These larvae released by copepods penetrate the human gut and enter the abdominal cavity and retroperitoneal space, where they mature into adult. The males die after copulation while the females migrate to the subcutaneous tissue. So this is called as subcutaneous parasites. Then sorry, then human get infected by this drinking contaminated water containing these copepods, and these contain larval three stage, which are released in the human body as soon as copepods die. The larva penetrate the duodenal wall and enter the retroperitoneal connective tissue, where they develop into mature adult male and females, and the females migrate to the host skin and causing severe discomfort and impairment. If you go to this life cycle of this larva Dracunculus, first when human drinks unfiltered water containing these copepods with inside the copepods, this larva completes its life cycle and with L3 larval stages, and this human become infected by drinking unfiltered water containing these copepods, infects with larva this Dracunculus. Following ingestion the copepods die and release the larva, because in stomach the stomach acid digests the copepods and release the this Dracunculus larva, mm, which penetrates the host stomach and intestinal wall. This Dracunculus penetrates through the host stomach and intestinal wall to enter the abdominal cavity and retroperitoneal space of the human beings. then they mature into adults and copulate, after which the male worms die and females migrates into the subcutaneous layer of the skin, and approximately 1 year after infection the subcutaneous tissue um, towards the skin, generally on the distal lower extremity, which rupture. This ulcer will rupture when the lesion comes into contact with water. The female one emerges and release to larva from this ulcer. This female larva rupture and release, and the larva are ingested by this copepod. Then this larva is released and this larva is ingested by this copepod, and after 2 weeks and 2 molts, after 2 molts it converted to infective larva, that is L3 larva. This ingestion of the copepods completes these cycles. And if you go to the pathogenesis, this guinea worm infection is asymptomatic for about a year. After a year it causes some ulcer of the skins, and which it causes symptoms, sorry, ulcer in the skins, and symptoms includes fever, nausea, vomiting and diarrhea, and itchy rashes, and the blister formed by the allergic reaction. This blister are also caused by allergic reaction of toxin released by female adults and causing a burning pain. Without adequate care, the wound get bacterial infection. This wound get bacterial infection, which can result to formation of abscesses. Abscesses can also occur when the worm migrate to the other tissue like lung, pericardium, spinal cord. This bacterial infection also causes redness, swelling in the skin, cellulitis, sepsis, joint interaction, and paralysis, and even tetanus. This allergic reaction also causes this uh, disease called. If you go to the next detection page, we also detect through microscopic examination of this species, and also through the immunodiagnostic like ELISA methods, and the detection of immunoglobulin G4 variant 4. IgG G4 means IgG isotypes. G4 appears to the most specific response. IgG is specific to this Dracunculus species, and the test is used for detecting pre-patent infection up to 6 months before they arise. Before 6 months, we cannot detect this parasite species. Then, if you go to the immunity and immune evasion mechanism, pathogen can evade immune response in several ways, including antigenic variation. If they if they changes this antibody, uh, immunoglobulin G4 express for this particular antigen of Dracunculus species. If this species changes or varied this antigen, then this antibody will not get activated, and the it evade the immunity of the human beings, and resistance to immune effector mechanism and suppression of immune response. Then it will cause suppression of immune response. If we then go to the treatment and drug targets, the infected individual should care the wound, because wound, if we not care the wound, the bacterial infection increases the Dracunculus species. To avoid this infection of the open ulcer while the worm is living, once the worm begins to exit the body, the worm should be cleaned, applied with an antibiotic ointment, because antibiotic ointment kill this bacterial infection. Uh, uh, and this species is begins to exit, and wrapped with a fresh gauze, and the antihistamines and steroid can help the early stage of allergic reaction and painkiller, because first stage of infection causes allergic reaction in skin. So we treated with different types of antihistamines and also in different steroids and also with antibiotic ointments. Then if we go to the different vaccine strategy, then there are no approval therapeutics or vaccine available for this dracunculus species. Then nowadays proteomic and genomic investigation have proposed a development of a multi-epitope vaccine. One of such examples is multi-epitope vaccine designing using T cell and B cell epitopes, because it they changes the antigenic variation for T cell and B cell. So T cell and B cell will not detect this antigenic variation of this species. So we have to design these changes of these epitopes for Dracunculus species proteins, and also and different domain like lactamase B domain containing protein, G domain containing protein, and ferrochelatase. In addition to the adjuvants and linkers, however, research is still in early phases. For this species, research is going on for because of this antigenic variation. So we completed all the parasites, monogenetic and digenetic parasites. କାହାର କ'ଣ ଡାଉଟ୍ ଅଛି ଯଦି କୁହ। ଡାଉଟ୍ କାହାର ଅଛି? ଡାଉଟ୍ କାହାର ଅଛି କି?
No, sir.
नहीं सर आगो को चलो
Nice, sir. Good evening, sir.
ଗୁଡ୍ ଇଭିନିଂ।
Sir, ଆଜି ଆମର ଲାଷ୍ଟ କ୍ଲାସ୍ ଅଛି କି ପାରାସାଇଟୋଲୋଜିର?
ନା ମୁଁ ସାର୍ଙ୍କୁ କହିବି ଆଉ ଗୋଟେ କ୍ଲାସ୍ ନବା ପାଇଁ।
Yes, sir. ଆପଣ
ଯେହେତୁ
ଆଉ
ମୁଁ
ଗୋଟେ
ଆଉଟ୍ ଅଫ୍
କ୍ଲାସ୍ ନେଲେ ଆମେ ରିଡ଼ିଂ କରିକି ଆପଣଙ୍କୁ ଡାଉଟ୍ସ ଗୁଡ଼ା କହିଦେଇ ପାରିବୁ।
ଠିକ୍ ଅଛି, କାହିଁକି ନା ମୁଁ ଟିକେ ଆଉଟ୍ ଅଫ୍ ଷ୍ଟେସନ୍ ଅଛି, ତ ସେଥିପାଇଁ କି ଆଉ ଗୋଟେ କ୍ଲାସ୍ ମୁଁ ସାର୍ଙ୍କୁ ରିକ୍ୱେଷ୍ଟ କରିକି ନେବି।
Okay, sir. Thank you, sir.
ଓକେ, ଠିକ୍ ଅଛି।
नहीं
ମୁଁ ପିପିଟିଟା ପିପିଟି ବି ମୁଁ ପିପିଟି ବି ସେୟାର କରିଦେବି ଆଜି। ପିପିଟି କି ଥରେ ରିଡ୍ କରିକି ତାର ଭଲ୍ୟୁମ୍।
Yes, sir.
ରେଣୁବାଳା ଦାସ,
Yes, sir.
ରେଣୁବାଳା ଦାସ, ପ୍ଲିଜ ସାଇଲେଣ୍ଟ ୟୋର ଫୋନ। ରେଣୁବାଳା ଦାସ,
वाला मैडम म्यूट करंतु
ସେଇ କଥା କହିଲେ ତୁ ଖାଇଲା ବେଳକୁ ତୁ କହ ତୁ କୋଉଟା ଖାଉଛ ନ ଖାଉଛ ଆମେ ଜାଣିବି କେମିତି? ଇଏ ସରୋଜିନୀ କହିଥାନ୍ତା ରାନ୍ଧି ସାଇଲାଣି, ତୁ ସରୋଜିନୀ ଟା କହିବୁ ନା ଖାଉନାହାନ୍ତି ବୋଲି ଅଲଗା ତରକାରୀ ରାନ୍ଧି। ସିଏ କହିଲାନି, ସିଏ
वाला
ତାର
मैडम
ସୁଧୁ ଖାଇଲାନି।
म्यूट करंतु
ଠିକ ମାନେ ଖାଇଲାନି କେମିତି ବସିଲା ତା ରାସ୍ତା
ପ୍ଲିଜ କିଏ
ଖାଇଲାନି।
ଯଦି ଥିବେ ରେଣୁବାଳା ମ୍ୟାଡାମକୁ ଫୋନ କରି କୁହନ୍ତୁ ମ୍ୟୁଟ କରିବା ପାଇଁ। ସେ ଏବେ କଲେ। ମୁଁ ପିପିଟି ଟା ବି ସେୟାର କରିଦଉଛି ଆଜି, ଗୋଟେ ରିଡ୍ କରିଦିଅନ୍ତୁ। ଆଉ ନେକ୍ସଟ କ୍ଲାସରେ ଯୋଉ ଯୋଉ ପାରାସାଇଟ୍ ଆମେ ବି ଡିସ୍କସ୍ କରିଛେ ତା ଉପରେ ଯଦି କିଛି ବି ଡାଉଟ୍ ଥିବ ମତେ ଲାଷ୍ଟ କ୍ଲାସରେ ପଚାରିବେ। ମୁଁ ସାରଙ୍କ ପରମିଶନ ନେଇକି ଆଉ ଗୋଟେ କ୍ଲାସ ନେବି। ଠିକ
Yes,
ଅଛି।
sir.
ଓକେ
ହଁ,
ସାର୍।
Yes, sir.
ओके सर
Okay, sir.
ଆଜି ଏଇଠି ଆମେ କ୍ଲାସକୁ କ୍ଲୋଜ କରିବା।
ଓକେ
थैंक यू सर
Thank you, sir. Thank you, sir.
ଓକେ।
Thank you, sir. Thank you, sir.
ক্লাস করলে বলো না
ସାର୍ ସାଉଣ୍ଡ ଆସୁନି ସାର୍। ହ୍ୟାଲୋ ହ୍ୟାଲୋ